So today, our speaker is PhD student Qingyu Wang. Qingyu is currently a second-year PhD student at OU School of Meteorology, which works with Dr. Sean Crowell on CO2 flux and transport and helping to evaluate a couple of mesoscale, atmospheric, and biospheric model, or the WRF-VPRM by Dr. Xiao-Ming Hu. She attained her bachelor's degree at Nanjing University in China in 2016, where she did some research on observation of particle structures and variations inside precipitation systems by using C-band dual-polarimetric radar data. In 2019, she earned her master's degree at OU, working on atmospheric variations in column-integrated CO2 over the US. Today, she'll be speaking to us about greenhouse gas comparisons between the WRF-VPRM and tower observations at the urban scale. And with that, I'll let you go ahead and take it away.
Thank you, Liz. Thank you all for being here. I hope you all are going well. So my topic today is greenhouse gases comparison between WRF-VPRM and tower observations in urban scale. This is a PhD update, and I will have a long way to go after that. Dr. Xiao-Ming Hu and my advisor, Dr. Sean Crowell, worked together with me on this project. Okay. Oops. First, I will introduce our motivation, like anthropogenic emissions contribution, and why we use models to simulate it. And data we are using are tall tower measurement and WRF-VPRM simulations. Thereafter, we'll have some results and conclusions. Okay, let's first go to first part. The urbanization and climate change are two defining environmental phenomena of the 21st century, and these two processes are increasingly interconnected, as rapid urbanization is often accompanied by a change in like lifestyle, increasing consumptions and energy uses, which contribute heavily towards climate change. Okay, so moreover, as centers of economic activities and population migration and energy consumption, urban areas play a very significant role in tackling global climate change. Estimating CO2 emissions at the urban scale is therefore the first step for adaptation and mitigation of climate change by local government. But it is also hampered by the absence of comprehensive and reliable collected data on urban energy consumption and the related emissions. However, estimating urban CO2 emissions are very difficult due to reasons like the definition of urban areas. It is very hard to define the boundary of the urban area, and the buildings of human activities also makes the boundary layer in a city more complicated. Also, there are no measurement in the urban scales in urban areas. So the figures I give here, the first one is the traffic in a city. So the cars in the city contribute a lot of fossil fuel emissions in a city. And the second one is a dog in a traffic jam. The poor dog has waited for 1 hour in the traffic jam and he got so bored. The third figure is an interesting project I found online. It is called Zero Emission City Project. So in this project, members work together to jointly drive concrete CO2 emissions reduction projects that helps city get on a pathway to zero net emissions. They plant a lot of trees to absorb the CO2 contributed by the anthropogenic activities. And the last figure is fossil fuel emissions produced by a lot of factories, which is very scary. Okay, so to develop and assess methods of quantifying greenhouse gases emissions at urban scale and their role in larger scale greenhouse gases transport. A lot of observations and model simulations can be used. The first figure is a global observation site which observing not only greenhouse gases, but also like radiation and ozone and aerosol and meteorology and a lot of others from like surface sites, tall towers, airplane, ship, and so on. We will focus on tall towers measurement only in this case. Okay, so tall tower measurement allow us to determine the flux. What I mean by flux is how much it is being released or soaking up of trace gases over larger areas than instead of just measuring at the surface. The figure 2 is an example of the tall tower, which is in Park Forest, Wisconsin. This tower has 6 levels. The tallest level is about 400 meters above ground. So when and the tower measures CO2 and carbon monoxide, CO. When the boundary layer is shallow, which might be 300 meters or 200 meters, the tallest level make it possible to directly measure CO2, CO, meteorology, and turbulence in the boundary layer. Okay, so the tall tower network provides regional representative measurements of carbon dioxide and the related gases in the continental boundary layer. Measurements began on tall towers in the 1990s in order to extend long-term carbon cycle gas monitoring to continental area. So they uses existing television, radio, cell phone towers to be the sampling platform for in situ and the flash sampling of CO2 and other atmospheric trace gases. In this first figure, I give some red stars to mark the tall towers and the surface sites in the continental US. Some sites like WGC and LEF and AMT, they are all located in forests. And sites like SGP and WKT are on grassland. So the different locations of those tall towers allow us to have samples from various land covers. Okay, this figure in the lower panel shows measurement areas of the tall tower compared to the surface sites. Because air mixes upward over time, the higher you sample the air, the larger footprint you are sampling from. The black arrow you see in the figures represent actual distance, and the area under the green footprint circle represents how large an area influences the air being sampled. Tall tower can sample the atmosphere that is representative of a much larger footprint compared to the sampling air at the surface. So this is one of the benefits to have tall tower measurement. Those tall tower measurement can continuously sample greenhouse gases and a lot of variables we need because they can monitoring a larger area compared to the surface sites. Okay, so this figure are the land covers of the continental US. If we compare this to this sites, we will find so a lot of sites are over a lot of different land covers. And then we go to for VPRM. This is a model we're using simulations from. Okay, so this flowchart briefly reviews how WRF-VPRM works. It uses meteorological data, including terrestrial data and graded data, and chemistry data as well, which is from Geoschem. So they are serving as inputs into the WRF-Chem model, and the shortwave radiation and surface temperature from the WRF-Chem are applied together with modest satellite surface reflectance and the flux tower measurement. So they will produce the GE and the respiration. GE, which is gross ecosystem exchange, which is simulated from VPRM. VPRM is an aberration of vegetation, photosynthesis, and the respiration model. So from this model, we all have, in this way, the WRF-Chem and the VPRM are one-way coupled. So then from VPRM, we also have CO2 biosphere fluxes. Also, we can use oceanic emission from Takahashi and the fossil fuel emission from ODAC. Then we put those three fluxes into the carbon tracker model. Finally, we'll have the biogenic, anthropogenic, and oceanic CO2 concentrations. If we combine these three together, we will finally have total CO2 concentration simulations from WRF-VPRM. So this model is performed by Dr. Xiao-Ming Hu, and he gave this structure of this flowchart to me. Okay, then we go to the results. Since we have sites spreading over the US, I grouped some sites which have similar land covers together. So these three sites are from forest sites. The first one is at Park Falls, Wisconsin. So that black scatters are tower observations of the CO2. And the red scatters are modeled CO2, which is a combination of anthropogenic and the biogenic and the background as well. And the green shadow is also produced by modeled CO2, modeled WRF-VPRM model CO2, and the green shadow only consider the biogenic emissions and the background. And these left-hand side panels are the corresponding land covers at each site. Okay, so like for the first site in Park Falls, the dominant land cover is mixed forest and a little bit deciduous forest, grassland, savannas. And this site is at Argo in May. It is very similar to the first one, which has a lot of mixed forest and a little bit others. The third one is in Shenandoah National Park in West Virginia. But this one is a little bit different. It has most deciduous forest and a little bit mixed forest. So if we look at all the three figures, we can see there is a very strong seasonal cycle because of I think that is because of the land cover, because a lot of trees in the forest, they will absorb a lot of CO2 because of photosynthesis process. So CO2 in the summertime is less than other seasons. And if we look at the Shenandoah National Park site, the CO2 is becoming smaller. CO2 value is becoming smaller beginning at late April, which is earlier than other two sites. So we have two hypotheses for that. One is the Shenandoah National Park site is to the south of others, which has higher temperature in spring, which makes earlier growing season. And another hypothesis we have is they have different land covers, like deciduous forest maybe have earlier growing season than mixed forest. Generally speaking, I'm sorry, so I've got to say, so those plots are all at daytime in the afternoon. So generally speaking, WRF-VPRM matches observations well in the daytime. And WRF-VPRM matches also well on some synoptic weather patterns like here in the winter and like some cases in the summer. But even though the WRF-VPRM simulate well in the daytime, if we go to the diurnal cycle, this is a diurnal cycle by month at Park Falls, Wisconsin. So in each figure, the green line with gray shadow is the observation from tower and with the standard deviation at each hour. And similarly, the red line with a shadow is also the CO2 concentration from WRF-VPRM with standard deviation. This orange line is the wind speed at 10 meters high above ground level, and this blue line is the temperature at 2 meters high, and this green line is PBL height, and which units are 100 meters. If you see 5 in this y-axis, it is actually 500 meters above ground level. So all this orange line, the wind speed, the temperature, and the PBL height are from the model. And this blue box I mark here is the hours I included for the daytime plots in the previous slides. So it is from 15 to 19 local time. Okay, so from those figures, we can see that the model, the CO2, the red one, has a reversed pattern with the temperature. The temperature is also from the model. So we guess this is because the respiration function in the model is proportional to temperature. So the CO2, the biogenic emission part is depends on temperature, and also PBL height is related with radiation, and the radiation is also related to temperature. So the PBL height pattern is also similar to the temperature change, and which is very noticeable that in the summertime we can see a lot of very apparent seasonal diurnal cycle in a day, but in the winter time the CO2 concentration is very flat over a day. And also the model simulate the underestimated CO2 in the daytime, in the afternoon time. But if we go to the nighttime, the model is underestimated CO2, and the mismatch is very big. So we have some potential explanations to the mismatches. So one is because of the shallow boundary layer, and so the boundary layer is not well mixed as afternoon times. And another explanation is the imperfect flux in the model. Okay. Then we can look at grassland sites. The first site is at Lamont, Oklahoma. And the second site is at Moody, Texas. So both of these two sites are over grassland, but SGP has more cropland than WKT. Compared to the forest sites, we can see that there are not very strong seasonal cycle. The CO2 concentrations is kind of flat over the year, and it is quite noisy in the winter time and relatively flatter in the summertime. And in July, in the summer, at SGP, we can see that WRF-VPRM overestimated CO2. But if we go to the synoptic weather pattern, the WRF-VPRM matches that well in terms of variability, even though the model a little bit underestimated CO2 there. The difference between these two grassland sites is that SGP has more cropland than WKT. I think that's probably the reason why the WRF-VPRM overestimated CO2 in the summertime, especially in July. It goes to the diurnal cycle. Also, is similar to the former one. So the seasonal cycle, a seasonal variation of the diurnal cycle are the same with the forest. In the nighttime, the model underestimated CO2, but which is different with the previous one that in the daytime, the model a little bit overestimated CO2. And the winter time, the CO2 concentrations is also flat, which does not have the diurnal cycle as large as the summertime shows. And another kind of size is cropland sites. So I have two of them. The first one is at WBI, which is at West Branch in Iowa. It has a strong seasonal cycle as well as like forest sites. And Walnut Grove in California, which is in the valley of the California. It looks more. It looks like more like a grassland site, which has a weak seasonal cycle and a noisy CO2 concentration spreading in winter time. Even though the dominant cropland, dominant land covers are cropland, but these two sites are actually different. I search online and find the dominant crops in WBI is corn and soybean, but the crops in the WGC is much more complicated compared to WBI. So it has a lot of crops like grape, alfalfa, almond, walnut, winter wheat, and also this site is located at the valley of the California, so it is getting closer to the boundary. So it is driven by the boundary condition of the model. I found online that grapes, it has bud break starts in March. So I find interesting that starting in March, the CO2 concentrations is becoming flatter or concentrated. I'm sorry, it's more flatter starting in March, which is still noisy in the winter time. And the diurnal cycle is very similar to the grassland and the forest, which all underestimated the CO2 in the nighttime, but simulated well in the daytime or the afternoon. Then we have the last site, we'll call it a hybrid sites. So it combines, it includes savannas and mixed forest. So the dominant is savannas. I also include anthropogenic emissions in the first slide. So the anthropogenic emissions plus background is more than final CO2 concentrations from WRF-VPRM, but it cancels with the biogenic emissions from WRF-VPRM. And finally, we'll have the red dots, which is the CO2 from WRF-VPRM as a final results. So it has some features from mixed forest, which like it has a relatively smaller seasonal cycle compared to forest size, but a little bit stronger than the grassland size. And also, WRF-VPRM matches the synoptic weather pattern well, especially in the late August and the early September, and also the June cases. Okay. If we go to a diurnal cycle, we can say that WRF-VPRM simulated this site quite well. It's still underestimated CO2 at nighttime, but the mismatch is much smaller compared to other sites. And another interesting thing is there is also diurnal cycle, which is very apparent in the winter time. Okay. So in conclusions, WRF-VPRM simulates the seasonality well. So for some forest sites, they have a very strong seasonal cycle, and the grassland is flatter. And WRF-VPRM simulations matches synoptic weather patterns in time, like it matches synoptic weathers for the hybrid sites in August and September, but sometimes it is overestimated or underestimated. So, but it is not matches quite well in magnitude. WRF-VPRM also simulates CO2 well in daytime. So we have some potential explanations for that, like the biogenic and anthropogenic emissions in the daytime is working pretty well. And also in the afternoon, the PBL is well mixed, and this is better for WRF-VPRM simulations. But in terms of the CO2 simulation at nighttime, it is, for all the cases, it is underestimated, both in terms of mean and variations. The potential explanations for that is probably the first one is a poor flux representations, especially the biogenic emission. The respiration is proportional to temperature, so probably the function for the respiration is imperfect, and also the PBL dynamics in the nighttime is shallow, so it probably poorly modeled. For next step, we should include other timescales, like we should have diurnal, seasonal, and the synoptic timescales. We can focus more interesting cases in synoptic scale. We can also examine some meteorological data like temperature, moisture, and the PBL depth from real observed data, and also radiation from some surface sites. We can also compare the WRF-VPRM flux to eddy covariance data to help improve the biogenic and anthropogenic emissions, like that, to help improve the respirations and others. Okay, thank you, that's all.
Thank you, very good. So I went ahead and given everybody the ability to unmute themselves. So if anybody has a question for our speaker, go ahead and unmute yourself and chime in. Okay, well, I will hop in with one. I see people unmuting themselves. Sorry, go for it, Tyler.
Yeah, so I might have just missed this because my Wi-Fi was cutting in and out at the beginning, but what was the height at which you were comparing the surface obs to the model data?
Oh, yeah. So I mentioned that towers have a lot of levels. In those cases, I'm just using the lowest level. So it normally is like 30 meters above ground level, and sometimes it's 12 meters and 60 meters. So I'm always using the lowest level of the
Okay.
towers.
And what's the lowest level of the model?
The model is surface level.
Okay, thank you.
Mhm.
Could you go to the two crop cases?
Yes.
What did you hypothesize the difference between the oscillations was, or the yearly cycle? Was it due to were you hypothesizing it was due to the crops? Yeah.
So this, this one is crop, it is corn and soybean dominant, and their growing seasons in the summer. So it will absorb a lot of CO2 in the summer.
Oh, okay, sorry, I missed that.
But this case will have a lot of other crops like what I've given here. So they will have a lot of different growing seasons.
Okay, sorry, I missed that. Thank you.
Any other questions for our speaker today?
I have a question. Qingyu, you on some of your plots you broke it down. So the plot that we are looking right now, you have the red dots. It's anthropogenic, biogenic, and background, and then you have biogenic and background separated. Most of the time, the two lines agree very well, but sometimes there's actually a difference. Does that then, if the red line is above the green, does that indicate that the site might be influenced by some direct anthropogenic emissions, or how do we interpret those differences?
Yes. Sorry, I didn't include anthropogenic emissions with them. So like some case, I give the anthropogenic emission here. Yeah. So like this case, the anthropogenic emission plus the background is more than the red dots. But like here, the biogenic emission plus background is lower than the red dots. But the final result we use actually the anthropogenic plus biogenic plus background. So it will cancel. Another one I didn't include is actually the oceanic, but Dr. Xiao-Ming Hu told me that oceanic contribution is very little, so we didn't consider it. I'm not sure if it is actually contributing to the red dots. Probably in this case, the oceanic emissions is smaller than biogenic emission. So the three all cancel together, and finally we'll get this red dots.
Okay, and then I have a follow-up question. So you looked at this kind of breakdown when you look at the seasonal cycles. Do you think in looking at this breakdown, like what you're doing here, when you make the diurnal cycles, do you think that might provide you any information to what extent the differences that you're seeing at night might be due to issues with the emissions or with the boundary layer parameterization? So do you see value in breaking it down into these different parts like you did on the previous slides for these plots?
Yeah, I think I should go more to each case. Like this one is like hybrid sites, and I think this case matches the observations best in those cases I present But some cases, the mismatches are like we cannot say it is good estimation. So I'm actually not sure it is more big due to the poor modeled boundary conditions, or boundary layer dynamics, or the respiration function didn't work well. In WRF-VPRM, in the flow chart, the respiration is linearly proportional to temperature, and they have some like constants like those par or alpha, and it probably due to imperfect constants of those settings, and probably also due to the temperature simulation. So it's hard to say.
But you could, I mean, you could, in theory, you could go ahead and look at how the respiration data compare for the different sites diurnally, and whether you see differences.
Yes.
Between and also you could look at the CO2 fluxes, correct? The diurnal cycle. Yes. Okay.
I can do it as next step.
Thank you.
Mm-hmm.
Nice.